Hi there, and very welcome to this webinar on the ddPCR technology. And we will try to explain how it can be used in the research on parasites of livestock. My name is Johan Höglund, and I'm leading a group at the Swedish University of Agricultural Sciences. And we are working on helminths of veterinary interest and how they respond on anthelmintic treatment. For some years, much of our research has relied on various molecular applications, and in particular those based on ddPCR. In the presentation, I will first provide a general look back at the topic of molecular identification of GIN genera using PCR technologies. Then I will move on and give an overview of the principles behind droplet digital PCR technology and how it works, after which some of the advantages of the method compared to similar technologies will be discussed. This will then be followed by a brief presentation on the workflow used in ddPCR. And finally, I will go into how to proceed in terms of primer probe design and in the analysis of the data generated. The first question is how DNA-based tests can contribute to and improve parasite control in livestock. One vision is that the farmers will hopefully send in samples for diagnosis to a greater extent than that is happening today, and then only treat the animals when necessary. But this, of course, requires access to reliable diagnostic tools. Historically, the diagnosis of GIN has required the microscopic examination of fresh fecal contents for the presence of eggs following some kind of a flotation protocol. Although this is rather simple and easy to perform, it's well known that the identification of strong-eyed eggs is difficult, as most of them are more or less identical in form and shape. Accordingly, it's necessary to perform cultivation of larvae for differentiation. However, this requires access to fresh faeces, and this takes about 7 to 10 days to perform. Furthermore, as larval differentiation is based on minor morphological characteristics, parasitological expertise is required. But even then, the procedure is not always reliable due to culture bias. In contrast, the DNA-based tests are not investigative dependent and therefore more objective. But they also do allow for more sensitive distinction among the different worms present. So these tests provide farmers and their advisors with more confidence regarding decisions related to treatment and parasite control. Many of today's molecular tests also offer a promise of automated platforms, which saves time. And another advantage is that once DNA have been extracted, the samples can be stored. This provides opportunities to revisit the same samples, for example, if we need to screen them for mutations linked to anthelmintic resistance. I'm the first one to realize that we're not there yet for all drugs, but still, in the long run, this would be a major improvement. Since the advent of PCR in the early 1980s, a wide range of different molecular applications are broadly speaking now used for different purposes in clinical research. At the same time, it's important to emphasize that it was not until 30 years ago that the first reports became available in the research we are interested in. One of the first publications is this Australian study from Robin Gasser and colleagues in Melbourne, which already then suggested that PCR has considerable potential for the identification of eggs from different nematodes in fecal samples. I suppose this inspired many of us to embark on this track. However, ever since then, we have experienced enormous technical development in the field of molecular diagnostics. This image from Biorad nicely summarizes the development in the area. Although the PCR technology revolutionized the field, a well-known disadvantage of conventional PCR is that it's only possible to denote the presence or absence of various pathogens of interest. But this could be partially addressed a few years later by the invention of molecular probes and real-time PCR in the mid-1990s. But it was not until the launch of ddPCR in 2012 that a technical solution offering absolute quantification emerged on the market. Before I start to dig into the basic principles of the ddPCR technology, I just like to show the progress in the field of molecular detection of GIN in the faeces of ruminants. As can be seen in this graph, there has been a tremendous progress in the field, and with an ever-increasing number of publications. Still, to the best of my knowledge, none of these tests have as yet been included in the diagnostic portfolio in commercial labs. Furthermore, few studies take advantage of ddPCR, so there is still a long and winding road before these tests reach the stakeholders. Then to the principles of the technology and what makes it different from similar PCR approaches. Unlike real-time PCR, ddPCR is based on an endpoint approach that takes advantage of a water-oil emulsion droplet technology, which is combined with microfluidics. First, the reaction volume in each sample is divided into approximately 20,000 nano-sized droplets where the DNA content is spread randomly. Consequently, and as seen here, in the end, the contents of each drop are amplified independently as a separate PCR reaction. Ultimately, the results depend on fluorescence based on the droplet content in the sample. This is in sharp contrast to qPCR, which measures a certain cycle threshold level in real time. Now I intend to proceed to the workflow, which is performed in 3 separate steps, and where the samples are first loaded into one or more strips in accordance with the well-known 96 well format. The first step in the ddPCR procedure is the generation of the nano-sized droplets that partitions target and background DNA in each sample, as shown in the upper right corner. With the right equipment, this is done automatically. This will, of course, eliminate user-to-user variability, and it also saves time. The second step is to amplify the target sequence in each drop by endpoint PCR. After careful sealing of the plates with olive oil, this is done in more or less the same way as with conventional PCR. For more detailed information on PCR cycling steps, we refer to the Biorad user manual. Nevertheless, in the end, the droplets with template will emit the signal and glow, while those without will not react. Finally, the plates are moved to the droplet reader, and where the number of positive and negative drops is counted. The higher amount of DNA in the droplet, the stronger is the signal, as indicated in the figure in the upper right corner. Ultimately, this gives an exact quantification of target sequence in each sample. Again, this is done automatically, and the results can easily be viewed and analyzed with a software called Quantification. Here is a summary slide, again showing the different steps. So after mixing the samples containing the template of interest with a master mix with primers and probe, the PCR assay mixture are distributed into a very large number of compartments or droplets, such that there is either 0 or some target molecules present in any individual reaction. Finally, after amplification, the number of positive reactions gives the amount of template from which the concentration can be calculated. Then to how the quantification works. As pointed out before, more than one target molecule may be co-located in the same compartment, and where the number of molecules in each compartment follows a Poisson distribution. Thus, the number of copies of the target molecule in a sample can be calculated accurately according to the formula shown in the graph below. So far, I've only been through the basic steps behind the technology. Here I like to show different approaches that can be used for pathogen detectors or sequence variants. In principle, 3 types of assays can be designed. The simplest type is a Uniplex assay, where the DNA target is amplified using only one specific set of primers. This, like in any PCR application, where the amplified region usually is visualized by the use of hydrolysis probe that anneal within the target. During the enzymatic degradation of the probe with a Taq polymerase, it releases the fluoropore, when it comes in contact with the quencher denoted here by a black dot. The slight difference with the multiplex or duplex assay is that two targets are detected at the same time by the use of two different primer probe sets, but where each target is visualized by different fluoropores attached to each probe, in this case FAM and HEX. A third option is the so-called discrimination assay. In this case, the same template is amplified by the same set of primers. However, the sequence variants are distinguished in the same way as in the duplex reactions, but by the use of slightly different probes. In this example, the probes only differ in one position. In our lab, we have developed an assay for osteotagia and coperia detection, which is based on this type of assay. Interestingly, the discrimination assay is also well suited for detection on single nucleotide polymorphisms. For tips and tricks as regards primer and probe design, we again refer to the Bio-Rad application manual. What are then the major differences between qPCR and ddPCR? Firstly, qPCR is semi-quantitative as it relies on a certain predefined PCR cycle threshold, or the CT value. In contrast, ddPCR provides absolute quantification of DNA copies directly, since it relies on a PCR endpoint reaction. This without any need for a calibration curve. Secondly, in ddPCR, the issue of PCR efficacy is handled, and there are no problems related to DNA sharing or presence of inhibitors. This since the amplified templates are usually shorter as well as the reactions are divided and independent of each other. With the right equipment, the technology is also very flexible and user-friendly, since the droplet generation as well as the PCR and droplet reading steps are automated. Like with any PCR-based assay used for diagnosis of GIN, all the ddPCR protocols we have so far developed in our lab take advantage of the sequence variation in the internal transcribed spacer region 2 within the ribosomal RNA gene array. In the application shown here, we designed 4 sets of primers and probes targeting the 3 most important genera of strongyles that occur in sheep. To start with, 3 sets of genus-specific primers and probe were developed for Hemonchus, Telodoshagia, or Trichostrongylus in a variable region indicated with the red arrow in the figure. On top of this, we also designed a primer probe set situated a bit upstream in a conserved region shared by all genera, and which accordingly is universal for any strongylid worm. Before use, reaction conditions were carefully evaluated using a thermal gradient on DNA extracted from adult worms of each genera. This in order to avoid non-specific target reactions and to maximize the separation. So by multiplexing, we can now determine the proportion of each genera in ovine fecal samples. If you're interested in this or in similar applications, we refer to the publication that will appear in the end of the presentation. Then to the visualization of the results generated by the droplet reader and how they are shown in the QuantaSoft software. This figure shows a typical example of the one-dimensional plot with one set of primers and probe. Here, endpoint measurements in 7 samples, A to G, together with a non-template control to the right that are separated by yellow vertical lines are shown. Thus, the black dots are the negative droplets without target DNA, and as you can see, these are separated by a threshold from the blue dots containing variable amounts of target DNA. Ideally, the PCR conditions should be adjusted so there is a clear separation between the negative and positive droplets and with no rain in between. In fact, one beauty with ddPCR is that the threshold level in pink can be adjusted. I can assure that in reality it does not affect the outcome so much if the threshold would have been adjusted upwards. Nevertheless, to be sure of the baseline, one advice is to always include a non-template control in every run. This way, it's possible to play with the data and to exclude all data points that represent false positives. Nevertheless, with the original settings in this example, the first 3 samples contain no or very little DNA, whereas samples D to G contain large amounts. As outlined before, ddPCR also offers the opportunity to run different primer probe sets combinations, provided that they do not cross-react and interfere with each other. In this case, the results can be viewed either separately as shown in the previous slide, but it's also possible to view and analyze the results produced by both channels at the same time as seen here. In this example, we use our duplex assay for strongyles in sheep. The blue dots, they represent droplets that are positive for Haemoncus, whereas the green ones represent drops only containing the universal target. However, as you can see, on top of this, there is also a cloud of orange droplets that contain both targets. Based on this information, it's possible to determine the total number of strongyle DNA copies in a sample, but also the fractional abundance of Haemoncus. To sum up the advantages with ddPCR, firstly, it provides unmatched precision with the massive sample partitioning afforded. This enables small fold differences in target DNA sequence between samples to be reliably measured. Secondly, the method gives an increased signal to noise. This since ddPCR enrich for rare targets by reducing competition that comes from high copy templates. Thirdly, error rates are reduced by removing the amplification efficacy reliance of the PCR. This permits accurate quantification of targets. Finally, the quantification is absolute, but also simplified since a standard curve is not required. After listening to all of this, I hope you have now become convinced that ddPCR is a flexible and simple technology with wide application in our research field. Remember, not until these tests have been put in place we're able to quantify parasites correctly, which of course is important for the implementation of sustainable parasite control strategies. Thanks for your attention.